Welcome to Azad Bioacademy. Today's lecture topic is mechanism of insulin. That means how insulin works within our body to lower the blood sugar and other function. Okay, so no delay, come to the main point, that is mechanism of action of insulin. Okay, so insulin is a hormone which is secreted from the pancreatic beta cell, and that insulin will work on a receptor. That means it will work receptor oriented. Then that receptor is basically kinase, that means enzyme linked receptor, tyrosine kinase receptor. Okay, so insulin comes from pancreatic beta cell, and when you will take carbohydrate from your GI tract, a chemical signal by incretin, GLP, GIP like, that will reach to your pancreas, and that will activate the beta cell to release the insulin. Alternatively, when you take carbohydrate, that will break down into a smaller molecule, that is glucose, and that will enter into the blood, and through blood it will reach to the pancreas, and pancreas will release insulin, and that insulin will work to lower the blood sugar. Okay, so firstly, already I said, that is insulin work on the receptor to lower the blood sugar level. So firstly, I will just draw a membrane, and before going to the figure, just I want to add something. Insulin works on insulin receptor to lower the blood sugar level, but not all the tissues required insulin to take glucose. Okay, like your skeletal muscle, your adipose tissues, and your cardiac muscles. These cells require insulin to take sugar. Other than, you can think about your brain, your lungs, your kidneys, has no need to insulin to take blood sugar. Okay, so insulin work on receptor, and the insulin receptor is made up of two monomer of the polypeptide. So one monomer polypeptide is like that. Okay, so this is one monomer chain polypeptide, and this is another one, and this extracellular part is alpha subunit, and intracellular part is beta subunit. That means insulin receptor has alpha and beta part, and this receptor is transmembrane receptor because it's already crossed the membrane. The beta portion is just inside the cell, and alpha portion is outside the cell. Okay, so this is insulin receptor, and now insulin will work on it. So this is, you can think, this is insulin. So firstly, that insulin will bind with the alpha portion, and that insulin bind with that monomer part of the alpha portion. Yeah, you can think, single receptor, and it will be activated by a single insulin. Actually, not like that. It is somewhat different. That means two insulin will bind with the two alpha part of the two monomer chain, and it will change the conformation, and it will come very close to each other, and it will change its conformation, and that changing conformation, the beta portion will be activated. How it will happen? That is, if you just try to catch something by your hand, then you must just fold your finger. Okay, that is alpha part. And if you think your lower hand is beta, so when you try to catch something, your beta part is just stretching, and it will be activated. So how it will be activated in molecular level, that is the another question. So that question is very important. That is, inside the beta part has a particular enzyme, that is tyrosine kinase enzyme. That is tyrosine kinase enzyme. Okay, and the beta region, this is the polypeptide chain. So of course it is made up of amino acid, and it has some portion which is very much rich in tyrosine, and you can say it is tyrosine residue. Okay, so this is tyrosine. Okay, so when the insulin will just bind with the insulin receptor, and insulin receptor will just change this conformation to capture the insulin, and it will stretch the lower part, and its conformation will change, and it will activate the tyrosine kinase. And what is the function of tyrosine kinase? Actually, the kinase enzyme, the function of kinase enzyme is to phosphorylate something. Okay, so what will happen? That kinase enzyme, will phosphorylate on its own tyrosine residue. That means that tyrosine will be phosphorylated. Who will help to phosphorylate it? That tyrosine kinase enzyme will phosphorylate that region. So this is their own part, and it is phosphorylated by tyrosine kinase. So this is called autophosphorylation. Okay, without help of other molecule, it is phosphorylated by its own by the help of its own tyrosine kinase enzyme, which is intrinsic enzyme of the beta subunit. Okay, then what will happen? Already beta portion, the tyrosine residue already phosphorylated, and its changes structure, and it is completely active now. Okay, so this is very active residual beta part of the insulin receptor, and it is ready to work to activate the intracellular signaling pathway. Okay, then what will happen? That phosphorylated residual part of the beta subunit, it will recruit some molecule to activate the intracellular signaling pathway. Okay, and that molecule, you can think this is the molecule. This is protein, and that protein is called insulin receptor substrate protein. That insulin receptor substrate protein will be well matched to the activated tyrosine phosphorylated tyrosine part, because its domain will match to bind with it, because it changes its structure to bind with to recruit the insulin receptor substrate protein. Okay, so what will happen? The insulin receptor substrate protein will bind here. Okay, that means insulin receptor substrate protein is just being recruited by the phosphorylated tyrosine residue of the beta part of the insulin receptor. Okay, so what will happen now? Now this is completely attached with the residual part. Then this is insulin receptor substrate protein. So it is also made up of amino acid, and it has also a residual part of tyrosine. It has also tyrosine amino acid. Okay, then what will happen? That tyrosine will be phosphorylated by the tyrosine kinase enzyme, which is integral part of the beta subunit. Okay, that means it will phosphorylate. Phosphate will add. You can think autophosphorylation, and here the phosphate, where it will get phosphate to add the phosphate. Phosphate comes from the ATP converted to ADP, and one inorganic phosphate will release. Okay, and that phosphate will be attached there. So that is the phosphorylation. So when the insulin receptor substrate protein will attach, then it will be phosphorylated by the tyrosine kinase enzyme. Okay, so now what will happen, that insulin receptor substrate protein, which is already phosphorylated, that is, you can say, active insulin receptor substrate protein. Okay, that insulin receptor substrate protein will recruit or will activate the other kinase enzyme, that is phosphoinositidyl 3 kinase. And you can say it can also activate other pathway, that is mitogen activated protein kinase. But our concern is to lower the blood sugar, so we will go in that pathway. Okay, that is phosphoinositidyl 3 kinase pathway. So the insulin receptor substrate, I want to add something on insulin receptor substrate, that is insulin receptor substrate has some types, okay, based on different cell tissues. But here in skeletal muscle, in your cardiac muscle, in your adipose tissues, basically insulin receptor substrate, two types, insulin receptor substrate 1 and insulin receptor substrate 2. Okay, so both insulin receptor substrate can activate that PI3K, but mostly by inositol, in insulin receptor substrate 1. And then what will happen? That will be activated. It will be activated by IRS1 or 2. Then what will happen? Okay, it will be activated. So we can give a figure here. That is, you can think this is PI3K. When PI3K will be activated, then within your membrane has some substance, that is PIP2. That means phosphoinositidyl 4,5 bisphosphate. That means it is made up of glycerol, fatty acid, and inositol molecule. Okay, you can see this is inositol molecule, and over there glycerol and other fatty acid. So that glycerol molecule carry 2-phosphate. Okay, because of that it is called PIP2. That means phosphoinositidyl 4,5 bisphosphate, and that bisphosphate will turn into PIP3 by the enzyme this. That means PI3K phosphonosidyl 3 kinase enzyme will add one phosphate into the PIP2 and then it will turn into PIP3. So you can think this is your PI3. Okay, this is your PI3. So then what would be the function of PI3? PIP3? That PIP3 will now activate the one of the major molecule of this pathway, that is AKT, or you can also call it is protein kinase B. Okay, it will activate PIP3, activate the AKT. How it will activate? It will activate with the help of one enzyme, that is one enzyme that is called phosphoinositidine dependent kinase 1. Okay, when PIP3 will activate the PDK1 and then it will phosphorylate AKT. Okay, so what will happen? That AKT will be phosphorylated. So this is the AKT and one phosphate will be attached here. Okay, so this is the active AKT. So this is the master molecule of this pathway. How it is called master molecule of this pathway? Now we will just discuss. Okay, so you can draw the membrane in that way, the cell membrane. Okay, like that. That AKT will translocate the intracellular glucose transporter vesicle. So this is one vesicle which carry 4 glucose transporter. Okay, so this is called glucose transporter vesicle, and it must be translocated into the membrane to create a glucose transporter. So these are the glucose transporter. Okay, and this is the glucose transporter vesicle which carry the glucose transporter. Now what will happen? AKT here on substance is present. You can name it, that is AS160. You can also call this is GT page activated protein, that means GAP. Okay, so what is the function of AS160? That AS160 will just remove one phosphate from the ATP and it will make into ADP. Where it will work? So here another molecule is there, and this molecule name is called RAB. Okay, RAB protein. That RAB protein, before activation, RAB protein is attached with GDP, and during activation it will be attached with GTP. Okay, and when the GTPase activated protein will just make the RAB protein inactive. That means it will remove the one phosphate from the ATPase. Like this RAB protein is attached with GTP, okay, and it is active. The gap protein, what it will do? It will just remove the phosphate and it will make it GDP. So it will be inactive. Okay, so AKT, the function of AKT is to phosphorylate the gap protein. Okay, so this will phosphorylate the AS160. So when it will be phosphorylated, then it has no chance to make inactive the RAB protein. Okay, that means RAB protein will be active with the attachment of GTP. Okay, so it is very important to translocate the glucose transporter vesicle from the inside of the cell into the membrane, and who will just play important role for this? The master molecule is the RAB. Okay, this one. So RAB will translocate. RAB will just translocate the vesicle into the membrane. It will work alone or it will take some help. So with the help of motor molecules, some motor protein inside the cell, that will just translocate the vesicle into the membrane with the major role of RAB protein. That motor proteins are basically, the myosin 5, you can say kinin, you can say microtubules protein. That means different types of motor molecules, motor proteins are there. With the help of motor protein, the RAB protein will translocate the glucose transporter vesicle to the membrane. Okay, so come here. So that vesicular protein will attach with the membrane, with the transporter, glucose transporter. Okay, and it will fuse the membrane, and that transporter protein will be a part of membrane. Okay, so this transporter, glucose transporter, is called GLUT4. Okay, GLUT4 transporter protein. This is GLUT4, and vesicle already can become the part of membrane. So through this transporter GLUT channel, if you think this is glucose which is in your bloodstream, and it will enter into the cell through the GLUT4 channel. okay, and your glucose will be inside the cell from the outside. Okay, so from your blood glucose will enter and blood sugar level will be lower in the bloodstream. In that way, your insulin will decrease the blood sugar level. Okay, so to just to make the transporter protein translocation from the inside to the membrane, that means it's one kind of making a gate glucose channel. So usually we know we just superficially we heard there is insulin can open the glucose channel and glucose will enter from outside to inside. Actually, this channel is not like that ion channel. It is basically translocate. That means new transporter, glucose transporter will attached within the membrane, and through this transporter, glucose transporter, that is GLUT4, glucose will enter. Okay, and it will take few seconds, few minutes, very quick process. Okay, and what will happen if your sugar demand within the cell are not into the optimum level, then your sugar level will not enter, because demand must be created. Okay, so who will create demand? That glucose utilization will be enhanced by the AKT. Already I said that's AKT, activated AKT. This is activated AKT, is a master molecule. One of the influential molecule is RAP. So that AKT will increase the utilization. AKT will increase the utilization. So as we do not discuss the MAP kinase pathway, so I will just omit it. So this is AKT will also increase the utilization. How? It will increase the glycogenesis. It will increase the process of glycolysis. How it will increase the glycogenesis? That means glucose will make a glycogen inside the cell, and the demand of glucose will increase, and through this process glucose will enter. And glycolysis means the metabolism, a breakdown of glucose. Okay, so here some point. I will not go into the details, but I will just superficially I will just mention. That is, AKT will just work on the GSK 3 kinase enzyme. That means your glycogenesis pathway, that enzyme will work, and it will actually, this enzyme is responsible for inhibition of glycogen synthase enzyme. That glycogen synthase enzyme will be inhibited by that enzyme, by the phosphorylation. But what AKT will do? That AKT will phosphorylate that enzyme. Okay, so when that GSK3 will be phosphorylated and it will be inactive, then your glycogen synthase enzyme will be active and glycogen process will be enhanced. Okay, so indirectly AKT is just help to activate the glycogen synthase, and it will increase the process of glycogenesis. And another one is glycolysis. Glycolysis is a vast process, a metabolic process, and multiple enzymes are just needed to break down the glucose inside the cell. So here basically glucokinase enzyme, which will be activated by the AKT, glucokinase. Okay, and also other hexokinase, different types of enzyme are already concerned with that pathway. I will not go to the details already I said. So now insulin is working to enter the glucose from the bloodstream to the cell, and also it is increasing the demand of insulin inside the cell. That means it is increasing the utilization of sugar. So in that way, insulin will lower your blood sugar. So shortly, I will just sum up. So what will happen? You have alpha part of the insulin receptor. Insulin will bind the alpha part. Your beta part will be stretched, and within your beta part, tyrosine kinase enzyme over there, and tyrosine kinase enzyme will be activated, and your beta region, some tyrosine residues are there, and that tyrosine will be phosphorylated by the autophosphorylation process with the help of tyrosine kinase, and then autophosphorylated beta region, beta subunit, it will recruit the insulin receptor substrate protein, and that will be attached with the phosphorylated part, and it will be activated. And that activated insulin receptor substrate will just make two pathway. One is PI3K, another one is MAP kinase pathway. So our concern is that PI3K, that is phosphoinositol 3 kinase, and that will be activated by the insulin receptor substrate protein, and that activated PI3K will just make PIP2 into the PI3, and then PI3 will just activate the master molecule AKT. That means you can say also name is protein kinase B, and it will be phosphorylated by the help of one kinase enzyme, that is PDK1, and that PDK1 will be activated by the PI3K. Okay, so the AKT will be activated, and activated, that means phosphorylated AKT will play an important role to make an active RAB, because here is the enemy of active RAB, and that enemy of active RAB already I just erase it, that is your AS160, or you can say the gap protein, and that will be phosphorylated, and it will be inhibited, inactivated or suppressed, and your RAB protein will be activated with the GTP. Okay, and then activated RAB with the help of some motor molecules, motor proteins, the insulin, glucose transporter vesicle will be translocated from the inside to the membrane, and it will be a part of membrane, and the transporter, glucose transporter GLUT4, through this GLUT4 will be the part of membrane, and through this transporter, glucose transporter 4, glucose will enter from outside to inside, and it will increase the blood sugar level. Okay, and also AKT, that is PKB, which also activate the metabolic enzyme of the glycogenesis and glycolysis, and it will increase the demand of glucose within the cell. So that process will just take few minutes to few hours. Okay, so in that way, insulin will lower your blood sugar and increase the demand of sugar within the cell. So this is all about insulin mechanism. Thank you very much.